Hello, everyone. Thank you very much for attending my talk. My name is Dawei Song, and today I'm going to talk about quantification of cellular attractions and matrix degradation in 3D extracellular matrices. And here are the collaborators of my work, Asad Oberoi at the University of Southern California, Chris Chen's group in Boston University, the Symmetrics Company, Thomas Seidel at the Sandia National Lab, and Nicholas Hugenberg at Duke University. Cells are constantly pulling and pushing its surroundings, and the attractions exerted by cells play a very important role in many biological processes. For example, in tissue morphogenesis, attractions can be used to change the shape and spatial organization of the cells. And for cell migration, attractions can be used to control the speed and direction of migration. In addition to these structural and mechanical events, tractions can also trigger signaling pathways that promote basic cellular functions, such as the stem cell differentiation. When a stem cell is cultured on top of a 2D substrate, it can sense the stiffness of the substrate by applying tractions. Depending on the stiffness, the cell can differentiate into different cell phenotypes. So this is just one example of how mechanics can change biology. All of these examples are telling us that it's really important to be able to quantify cellular tractions. 3D traction microscopy is a robust tool to measure tractions in 3D. In a typical experiment, cells are fully encapsulated in the hydrogel matrix, and there are a lot of fluorescent beads embedded in the matrix. After a cell has spread it into the matrix and applied tractions, a set of microscopy images are generated to measure the shape of the cell and the location of the beads. Then the tractions are fully removed by killing the cell, and another set of images are generated to measure the location of the beads in the relaxed state of the gel. Then, making use of motion tracking algorithm, we determine the bead displacements in response to cell tractions. And finally, making use of the measured displacement, as well as the mechanical property of the gel, we can compute the tractions making use of a computational method. And in this talk, I'm going to focus on the last step of the workflow. Okay. Most 3D traction microscopy techniques make use of linear elasticity models to describe the mechanical property of the gel. However, these models are appropriate when the strain level is low, typically less than 10%, and when the matrix behaves linearly and isotropically. However, in real biological applications, none of these assumptions are necessarily true. In fact, experimental data has shown that the strains generated by cells can be really large. It can be as large as 50%. And most biological materials are highly nonlinear, so the shear modulus increases significantly as a function of shear strength. And a lot of these biomaterials exhibit a fibrous microstructure, and when you stretch the material, the fibers tends to be aligned along the loading direction, leading to a strong material anisotropy. And therefore, a nonlinear approach is required to accurately model the behavior of the material, okay? And on the other hand, the mechanical property of the gel are typically measured by standard mechanical tests, like shear rheometry and indentation testings. And these tests were typically done before encapsulating the cell, and it is commonly assumed that the matrix properties are uniform across the sample. However, when a cell spreads into the matrix, it produces enzyme that can locally degrade the gel, and thus inducing local heterogeneity in the gel. And in fact, experimental data has shown that depending on matrix composition, the degradation can be very significant. This is particularly the case when the matrix stiffness is high and when the matrix is highly susceptible to proteolysis. So therefore, it's really important to incorporate the effects of matrix degradation. Motivated by these observations, I'm going to show you how we can handle these challenges. So this is the outline of my talk. So first, I'm going to talk about the computational method, and then I'm going to show you 3 applications of our method. I'm going to show you how we can recover tractions in dextrin-based hydrogels and fibrous gels. And in these 2 cases, I'm going to focus on scenarios where the degradation is not very important. And finally, I'm going to show you how we can handle matrix degradation. So let's first look at the computational method. In this work, we formulate the problem as an inverse problem where our goal is to find the traction field T that minimizes the objective function. So here, the first term is a data mismatch term that describes the difference between a measured and predicted displacement field. And the predicted displacement field is required to satisfy the equation of equilibrium. And the second term is a regularization term that serves to ensure a certain smoothness of the traction field. So if you don't have this term, the reconstructed traction field will be very noisy and physically meaningless. So here is the computational workflow. For a given measured displacement field, we start with an initial guess for the traction field, and we solve a finite element problem to produce the predicted displacement field. And then we compare the predicted and measured displacements. If the difference is small, great, we're done. If not, we need to refine our traction field, and we repeat this process until the predicted displacement best matches the measured displacement. And we make use of a gradient-based method to refine the traction field, and we use an adjoint method to compute the gradient efficiently. Let's look at some results. So I'm going to first show you how we can recover traction in dextrin-based hydrogels. We are interested in these gels because they are relatively simple, and so they are amorphous gels and isotropic, but they exhibit nonlinear rheology that mimics the behavior of realistic tissues. So motivated by this, we make use of an exponential blast model to characterize the nonlinear behavior of the gel. So this model contains two parameters, mu and gamma, where mu denotes the shear modulus at infinitesimal strains, and gamma denotes the nonlinearity of the material. So you can see that a simple model can describe the shear rheology of this kind of gel. All right. So we first validate our method in silico. We start with a simulated traction field, and we solve a finite element problem to generate the displacement field. And then we add noise to it to mimic the realistic measured displacement field, right? And then we solve an inverse problem to recover the traction field. And finally, we compare the reconstructed and the simulated tractions, so the ground truth, to evaluate the performance of our algorithm. Okay, so when we solve the inverse problem, we use both a linear and nonlinear approach, and we want to see how sensitive the results are to the algorithm. All right, so here are the results. We can see that a nonlinear approach can fairly accurately recover the tractions in terms of both the root mean square of the traction and the maximum magnitude of the traction. However, a linear approach can generate large errors. So it can be as large as like almost 40%. So this means that neglecting the nonlinear effects can lead to significant errors in the traction reconstruction. And next, we apply our method to real experimental data. So the experiment will perform in Christian's lab, and this work were performed together with Symmetrics company. And we recover tractions for a total of 9 cells, and for each cell, we recover tractions using both a linear and nonlinear approach. And we want to see how different they are, okay? So here are the results. We can see that the tractions reconstructed by linear and nonlinear approaches are very different. So this reinforces our argument that neglecting the nonlinear effects can lead to significant differences in the traction reconstruction. So next, I'm going to show you how we can handle more complex gels like fibrous gels. So fibrous gels like collagen gels are commonly used to mimic a realistic cell environment. So these materials contain a random distribution of fibers in a background matrix. And motivated by this microstructure, we model the fibrous gel as a composite material, and we use an affine type homogenization model to describe it. So here you can see that the Cauchy stress has two contributions. The first term comes from the background matrix, so we use a New-Hookean model to describe it. And the second term contains contributions from the fibers over all orientations, okay? And we calibrate our model using experimental data for a uniaxial tension test. And you can see that our fibrous model can capture the nonlinear behavior very accurately. And for simplicity, we assume the initial fiber distribution is random. So here I plot the fiber density function. You can see that initially the fiber density is the same for all orientations, but as you stretch the material, fibers tends to be aligned along the loading direction as the fiber density increases significantly along that direction, all right? We also fit the experimental data using two simple phenomenological models, including the blast model that I just introduced and the new Hooking model. So the reason we do this is because we want to see if using these simple models is sufficient to accurately recover tractions. So we first validate our algorithm in silico. We start with a simulated traction field, and we solve a forward problem to generate the displacement field. So here I also plot the fiber density function for these 3 points. You can see that for a point that is really close to the cell protrusion, the fibers are strongly aligned, right? But as you move away from the cell, this effect is less pronounced. This is because the strain field decreases as you move away from the cell, right? So the alignment effect is significantly reduced. And so here are the results for solving the inverse problem. So we solve the problem using 3 models. So the first row shows the local traction error. And you can see that when a bad model is used, like a new Hooking model, the local error is really huge, okay? So this can also be seen from this figure. So this shows the normalized root mean square error for the traction field. You can see that if you use a new Hooking model or a blast model, the error is really huge. So this means the model error is significant, and you need to use a good model in order to faithfully recover the traction field. And finally, I'm going to show you how we can handle the matrix degradation. So to do this, we back up a little bit and we work with simpler gels like polyethylene glycol gel. So these gels are amorphous and isotropic. In addition, they are fairly linear even at large deformation, okay? So therefore, we can use a simple new Hooking model to describe the mechanical behavior of the gel. So here, the mu and lambda are the 2 Lamé constants for the gel. But because cells can produce enzymes that locally degrade the gel, so we treat these 2 parameters as unknowns, and we allow them to be spatially non-uniform. Okay, so here they can be written like this, and the mu naught and lambda naught, they are the original the original Lamé constants for a pristine matrix before degradation. So these 2 constants can be measured by a standard mechanical test. Okay, here this D is a degradation field that describes the extent of degradation. So our goal is for a given measured displacement field, we want to recover both the degradation field and the traction field, okay? We first demonstrate the feasibility of doing so. So we show that for a given measured displacement, we are able to uniquely determine the degradation field and the traction field. So we derived analytical expressions for them, all right? But these expressions work only when the measurement is perfect and doesn't contain any noise. Okay, so in practice, the measurement is of course always noisy, right? So we need to use an optimization strategy. So the idea is exactly the same as what I introduced before. The goal is to find the traction field and degradation field that can produce a predicted displacement field that best matches the measurement. All right, so exactly the same idea. And here are the results. So we validate our method in silico. So the first column shows the synthetic degradation field and the traction field. So motivated by experimental observations, we assume the degradation is most severe close to the cell surface, and it decreases progressively as you move away from the cell, all right? And the second column shows our reconstructed results. We call it traction and degradation microscopy. You can see that our method can fairly accurately recover both fields. And the third column shows results if we neglect degradation. You can see that if we do this, we significantly overestimate the traction field. This is because we assume a much stiffer matrix at the cell surface, and therefore we arrive at a much larger traction field. Okay. So here are the conclusions. We have developed an efficient adjoint-based inverse approach to quantify both cellular tractions and extracellular matrix degradation. And we use accurate constitutive models to incorporate the complex behavior of the matrix. And we found inappropriately accounting for ECM mechanics can lead to large errors, and our algorithm can be directly applied to a standard traction microscopy experiment. Thank you very much for your attention.